Hi everyone, my name is Savannah Moore and I'm here with Dylan Neal, Spencer Haroldson, and Ryan Jarrett. Today we're going to talk to you about the recent efforts being made to reduce gluten content in wheat using the CRISPR-Cas9 complex. This is important because approximately 77 million people worldwide suffer from celiac disease, which is triggered by gluten.
Celiac disease is an autoimmune disease that affects people by damaging the small intestine upon consumption of gluten. The disease is caused by a variant of HLA-DQ1A and HLA-DQ1B genes. Mutations of these genes trigger an immune response to gluten, resulting in the symptoms listed. The disease was discovered in 2nd century AD by Arrhenius of Cappadocia, while the true effect of gluten on patients was discovered in 1940 by William Dick. Since the discovery, much research has been performed to find treatments and hopefully one day a cure for the disease.
Celiac disease, left untreated, can lead to various other health issues. Following a strict gluten-free diet to treat celiac disease can also have negative impacts on long-term health. Individuals with celiac disease may be at a higher risk for developing other autoimmune disorders and health conditions such as diabetes, multiple sclerosis, osteoporosis, anemia, migraines, and infertility, many of which result from nutritional deficiencies. To prevent these issues, low-gluten wheat could be produced so that individuals with celiac disease can still consume wheat and obtain all other necessary nutrients without being harmed by gluten. Due to the complexity and high copy number of alpha-gliadin genes targeted for the reduction of gluten content, traditional mutagenesis and plant breeding efforts have failed. However, CRISPR-Cas9 has been used to effectively reduce the gluten content in wheat.
Alright, so now we can talk a little bit about the big picture. What is the goal here? Why exactly would knocking out the genes coding for allergenic gluten proteins in wheat be beneficial? Well, first off, celiac disease affects approximately 1% of the world population, equating to over 70 million people across the globe. Furthermore, 6 to 7% of people in the US are gluten sensitive, meaning that they experience some sort of reaction to the protein. Current popular wheat flour alternatives, such as almond flour or rice flour, do not taste the same and require emulsifying additives such as guar gum or xanthan gum. As you can see, the current gluten-free industry in the United States is worth over $1 billion United States dollars, so the proteins that compose gluten are a pretty big deal.
Using CRISPR-Cas9, researchers have been able to successfully reduce gluten content in wheat by targeting the alpha-gliadin gene family. 2 single-guide RNAs were designed to target the main immunodominant peptide that impacts patients with celiac disease, with one being more effective than the other. Various tests were used in this study to determine the gluten content in each line of wheat produced, and if any were transgene-free. In some wheat lines produced in this study, immunoreactivity was reduced by as much as 85%. Transgene-free plants with reduced gluten content that have been engineered with CRISPR-Cas9 could then be introduced into traditional breeding programs.
This figure directly compares plasmid-based CRISPR-Cas9 delivery to RNP-based delivery. Plasmid delivery takes many more steps involved in transport in, out, then back into the nucleus. This long process of nuclear import and editing can cause the gRNA and Cas enzyme to remain active for longer than necessary. This can cause off-target effects, resulting in unwanted cuts by CRISPR in regions that were not a part of the original cut site. RNP delivery reduces the steps involved in nuclear transport and editing by assembling the gRNA and Cas9 enzyme before transfection. This creates a much shorter 3-step process that lowers the amount of time that the RNP complex remains active, reducing off-target cuts. For reduced gluten wheat, reducing off-target effects can ensure higher quality consumer products without causing negative impacts to genes involved in bread structure and taste.
This figure discusses the use of CRISPR to disrupt the alpha and gamma gliadins, which are the gene families that cause the immunogenic response associated with celiac disease. The goal of this figure is to disrupt enough of the genes families to prevent the immunogenic response and create a hypoallergenic wheat. So why would we use CRISPR to develop this hypoallergenic wheat? Although non-immunogenic wheat already exists, the gene editing used led to a decreased quality in the wheat. Using CRISPR to edit these genes would allow for an alternative approach to developing non-immunogenic wheat without compromising its quality.
Alright, so how would gluten-free wheat benefit society? Well, as we've already mentioned, the population of gluten-sensitive people across the United States and the globe is incredibly large. Gluten-free wheat would mean that zero processing would have to occur to produce gluten-free wheat starch, since the unprocessed flour could simply be consumed. Using CRISPR gene editing on allergenic proteins also raises questions about future applications of the technology. Could we use the same tool to eliminate the proteins in peanuts that cause peanut allergies? What about other food allergies, such as soy, milk, or eggs? The answer to this is an emphatic yes. Even further, what about applications outside of agriculture? CRISPR is an incredibly powerful tool, and we believe that using it to reduce allergenicity is very powerful and appropriate application. Here are our sources, and here is our project breakdown. All right, and so our question is, which gene family is being targeted for gluten reduction based on the studies that we've evaluated? Take a moment to think about your answer and pause the video if you need some more time. All right, so the correct answer here is B, the gliadin gene family. This gene family codes for gluten, which triggers the response in humans for celiac disease. Thank you all.